Hi everyone. Welcome to the Genetics of Evolution lecture. Today I'll be talking about DNA replication and RNA synthesis in particular. And yeah, let's just get started. Okay, so before we go into deeply DNA replication and RNA synthesis, I think we have to first understand the basic principles of transcription and translation. So the central dogma of biology is basically the picture shown on the right. And what it's saying is that basically DNA, which is the double-stranded molecule we are all familiar with, um, becomes RNA and then into proteins. So this main concept of DNA to RNA to proteins is kind of like the baseline of today's lecture. So the process in which DNA transfers info from to RNA is called transcription, and it's basically when the synthesis of RNA it uses information from DNA. There are different types of RNA that particularly do this, and messenger RNA, mRNA, you must have heard that term, is kind of like the main RNA that carries a genetic message from the DNA all the way to the protein synthesizing machinery. So once the info is carried from the DNA to the RNA, the next step is for the information to be translated into proteins, and that happens through a process called translation. Now, in translation, what happens is basically there is a synthesis of polypeptide, which is protein, and basically amino acids are paired together to form a protein. So, which amino acids pair to each other and which amino acids come after each other, the sequence of all of that is defined by the amino acid sequence. And so the differences between DNA and RNA are important to understand before we go deeper into what it means to transfer information from DNA to RNA. So DNA has deoxyribose as opposed to RNA, which has only ribose. And also, so those are the sugar differences. And DNA has the four nitrogenous bases that DNA has are adenine, guanine, thymine, and cytosine. On the other hand, RNA doesn't have thymine and it instead has uracil. Additionally, RNA is single-stranded as opposed to DNA, which is double-stranded, and we can see that in this picture of the central dogma of biology. Additionally, translation happens in ribosomes, which are proteins themselves, and they help support, they facilitate translation. And yeah. So the differences between transcription and translation in bacteria versus eukaryotes are quite evident. Bacterial cells or prokaryotic cells don't have the complex machinery that is found in a eukaryotic cell. For example, organelles aren't like membrane. The more complex membrane-bound organelles are not found in bacterial cells. And most importantly, the nuclear envelope or the nucleus is not found in the bacterial cell. So transcription and translation both happen in the cytoplasm. And on the other hand, in eukaryotic cells, transcription occurs inside the nucleus because the DNA is concentrated in the nucleus. And the DNA, the information is going from the DNA to the mRNA, and only the mRNA is exiting the nuclear envelope into the cytoplasm. So there is no DNA in the cytoplasm, except a few rare exceptions. And also, there is a process called RNA processing over here, and it is unique to eukaryotic cells, and I will be discussing that shortly in a bit more detail. But yeah, the main points to remember are that transcription and translation both occur in bacterial cells as well as eukaryotic cells. And here in eukaryotic cells, the difference is that there's RNA processing and transcription occurs inside the nucleus. Okay, so I've been talking a lot about how DNA becomes RNA, right? How the information is passed from the DNA to the RNA. What exactly does that mean? So what that means is that we know there are four Oopsies. We know there are 4 nitrogenous bases in the DNA template strand, right? Which are adenine, cytosine, guanine, and thymine. So each of these bases pairs with another base. So for example, adenine pairs with thymine, cytosine pairs with guanine, and etc. And so what happens is basically the mRNA, it pairs along with the DNA template strand just like DNA would, except everywhere there's thymine, there's uracil. So everywhere the mRNA has to pair with an adenine, it attaches a uracil because it doesn't have a thymine. And this process is called transcription. So this is where this red strand is formed, which is the mRNA or pre-mRNA in eukaryotic cells. And so mRNA or messenger RNA is kind of like the messenger. It's like the strand that takes the information inside the nucleus to the outside, to the ribosome. So it's transporting that information from inside to the outside. And an interesting theory to not theory, but like an interesting idea is that the DNA is very it's a really important molecule and must be protected, right? So the DNA is kind of protected in the cell, and a way to remember it is that the DNA stays inside of the nucleus, while the mRNA, which is not as important as the DNA, is the one that does all the dirty work, like taking the information to the cytoplasm. Okay, and then mRNA is read in just like DNA. mRNA is also read by the bases, by the nitrogenous bases, and each set of so it's read in triplets. So what that means is that each set of 3 bases is called a codon, and each codon represents an amino acid. And on the next slide, I'll show you guys a picture of nitrogenous base triplets and the amino acids they correspond to. But usually, but basically the triplet set of 3 bases will code for a amino acid, and these amino acids will join together to form a polypeptide, which will again go through folding and more complex processes to become a protein eventually, right? And also this process specifically from the mRNA to the protein is called translation, right? So RNA makes the strand from the template DNA strand, which is called transcription, and then the amino acids are coded for using the codons, right? The triplet set of mRNA in the process of translation. Okay, so here's the table I was talking about. We can see that. So there's a lot of interesting things to look at in this table. Firstly, I'd like to point that. Firstly, I'd like to point that in the top right over here we have 3 stop commands, right? UAA, UAG, and UGA. So what this means is that when this order of nucleotides, so uracil adenine adenine, or uracil guanine adenine, or uracil adenine guanine come, this signals the stopping, right? It signals that there's no more translation or transcription that must occur. There's the protein is done. So it'll be cut off and the protein, the polypeptide chain will wrap around itself and become a protein. And similarly, along the same lines, we have AUG, which is adenine, uracil, and guanine, which signals the start, or methionine. So methionine is an amino acid of its own, and all proteins start with methionine. Okay, so how to read this table is you basically look on the right or the left, or you actually look on the left first, and you figure out okay, so let's say we wanted to see CCA, okay? So cytosine, adenine. So we'd first look in the left side of the table, and we'd isolate this row, and then we join it with the C column, so we know it's in this box. And then since it's A, it'd be CCA. So cytosine, cytosine, adenine, which is one of the triplet codons coding for proline, which is an amino acid. Okay, cool. So another important thing is that we have a lot of triplet codons coding for the same so multiple triplet codons are coding for the same amino acid. And what this means is that when a mutation occurs, if for example the third codon, the third nucleotide is supposed to be a uracil and instead becomes a cytosine because of some coding error or some transcription error or translation error, we'd still get isoleucine, right? Because that's why it's called that's why those certain mutations are called silent mutations. So the fact that multiple series of codons or triplets can code for the same amino acid gives an opportunity for mutations to be not as harmful. And at the same time, when one nucleotide changes to something really, really different, so for example, if let's say we're looking at cytosine, uracil, uracil, and cytosine, cytosine, uracil, and this middle nucleotide changes from uracil to cytosine, instead of the end product becoming leucine, it would be proline, and that's a problem because if there is even this is where even one mistranslated or transcribed nucleotide can cause a huge difference, because when we don't have the right amino acid in the sequence, it won't make the right protein, and the protein will be dysfunctional, and yeah, it'll cause problems. Okay, so diving deeper into transcription, we know that transcription. Ooh. So looking deeper into the first step of transcription, there's initiation, elongation, and termination. So these three steps also occur in translation, and these are basically the main points throughout the process where change occurs. So transcription again is the DNA-directed synthesis of RNA, where the RNA is synthesizing according to the DNA template strand. And what is initiation? Initiation is when the RNA polymerase, which is a protein, attaches to the promoter sequence on the DNA template strand. So I don't think you can see, but if you can, it's kind of green over there on the DNA template strand, and that's indicating the promoter. So the promoter will cause the RNA polymerase to bind there, and as soon as the RNA polymerase binds there, that means that initiation has occurred. Then the DNA unwinds because of a helicase or an enzyme that is specifically known for unwinding DNA. It comes and starts to unwind the double-stranded DNA, and the RNA transcript then attaches to the unwound DNA, so the separated DNA, and it starts to make its RNA transcript. So the mRNA is being formed. And an important thing to note here is, as soon as this is starting, right, the RNA transcript starts to make the mRNA, elongation is occurring. So elongation is basically the continual process of RNA transcript or mRNA forming as the DNA is being unwound and then rewound once that sequence is already used to make the mRNA. So as we can see over here, once the promoter sequence is, right, we started over here, and the RNA made the corresponding the RNA transcript made the corresponding RNA to this template DNA. So it unwound it over here, but now it again wound it again, rewound it. So this RNA polymerase keeps moving along the DNA template strand. As soon as it reaches a stop codon, right? As soon as it reaches the we talked about a few stop codons, right? UGA, UAA, and UAG. So as soon as it reaches one of those, it's going to stop. It's going to go off, and the completed RNA. Oh, excuse me. Actually, that the stop codon is actually supposed to be. These are stop codons corresponding to on RNA. So this is when translation occurs. But there are separate stop codons for DNA as well. And so the completed RNA transcript is released, and the last area where the RNA polymerase was will be rewound. And the direction of transcription here is downstream. So this is the most efficient way of transcription. It's also the direction from 3' to 5'. But the other upstream direction of transcription can also occur, but it is kind of less efficient. It's done in Okazaki fragments, and it's like the transcription is done, and then the RNA comes off, and then it goes back, and then again. So it's kind of like a repeated process with a lot of percentage of error. Okay, so I know over here, like this previous slide didn't have that much of a good picture, but over here I found some really good pictures of initiation and elongation. So here's the promoter that I was talking about. So the green part is the promoter, and in the beginning of every promoter sequence of DNA, there's something called the Tata box, which is basically a bunch of thymine and adenine nucleotides, and these basically signal the start of the promoter sequence. So yeah, um, the promoter and then the start point and the RNA polymerase. So before the RNA polymerase actually attaches, there are things called transcription factors, which are proteins which attach first. And the proteins attach and call for the transcription initiation complex, which is the RNA transcript. So, once the transcription initiation complex forms, the RNA transcript starts and it goes from the 5' to 3' direction. And during elongation of transcription, the newly made RNA is bound to the DNA, and I like this picture because you can clearly see that which nitrogenous bases are attaching to the DNA. So for example, remember how the thymine is not present in RNA? So we can see that here because the uracil is binding to the adenine because there is no thymine. So the red again is the RNA nucleotides, and the blue are the DNA nucleotides. And again, this is the efficient way of transcription in the downstream direction. Upstream is also done, again, that's in Okazaki fragments. Okay, so I know a couple of slides ago I mentioned RNA processing or RNA splicing. RNA processing is a special procedure that only occurs in eukaryotic cells. So this only occurs in cells that have a nucleus, and this specific process occurs inside the nucleus. And what this does is that the pre-mRNA, right, whatever has formed, transcribing to the DNA, RNA splicing will change that, will package it, will add some protective gear to make sure that this RNA can survive the conditions outside the nucleus. Because eventually, this RNA has to carry the information that it got from the DNA and send it all the way to the ribosome in the cytoplasm. So, in order for this RNA to make that, what I guess you could say, trip, or yeah, to survive the transportation, it must be given protective gear. So the first thing that is added is the 5' cap. So the modified form of guanine nucleotides, which are added to the 5' end of the first 20 to 40 nucleotides. So the 5' cap is basically a bunch of guanine molecules which are added to the front or the 5' side of the mRNA. And they're added to protect the mRNA. A similar function is exhibited by the poly A tail, which is as tail indicates is attached on the end, or the 3' side, and it's usually 50 to 250 adenine nucleotides. And again, this also serves as protection. And another really important thing that RNA splicing does is it removes the segments of RNA that are not going to code for amino acids. So the RNA will transcribe all the information from the DNA, right? It's not going to pick and choose, okay, I'm going to transcribe this part, I'm going to leave this part, and I'm going to transcribe that part. It's just going like a straight train and transcribing from the start codon all the way to the stop codon. So in that sequence, not everything can code for an amino acid molecule. Not everything out there really gives an end product. So in order to clear things up a bit, RNA processing or splicing, it removes the introns, or the segments that intrude the pre-mRNA, and it excises them together, so it puts them together and gets rid of them. And then so once the introns are removed, we are left with the exons, which are eventually put together or spliced together and are expressed. So we remember this is our exons are expressed, which is why they remain in the mRNA, while introns intrude, which is why they are banished out of the mRNA. And again, RNA processing, this fancy procedure, is only found in RNA in eukaryotic animals. And the benefits again are to facilitate that export, right? We want to make sure that the mRNA goes all the way from the inside of the nuclear envelope to the ribosome, so that it can actually make that it can actually code for that amino acid sequence, which will make the polypeptide chain eventually fold into a protein. Second important benefit is the protection from degradation. So from hydrolytic enzymes. So there are a lot of acidic molecules, or molecules that can basically hurt this RNA. And again, this offers protection. And additionally, what is the end product of this mRNA? It has to be able to attach to that ribosome so that it can make the polypeptide sequence, right? So in order for that attaching to occur, this 5' cap and poly A tail work together to help that ribosome attach to the 5' end of the mRNA of the ribosome. Okay, so now we finally come to the ribosome. So what does this mean? Ribosomes should spark the word translation, because the most important process that occurs in the ribosome is translation. Now, ribosomes are basically divided into two subunits. They have the large ribosomal subunit and the small ribosomal subunit. And there are three main steps in translation: the initiation, the elongation, and termination. And so again, translation is the RNA-directed synthesis of a polypeptide, because we are using the information from the mRNA to make the polypeptide sequence, right? So what happens first? So first, in initiation, here we see that this is the mRNA sequence, right? The start codon attaches. The small ribosomal subunit attaches at the mRNA binding site, and the initiator tRNA comes along and binds the methionine. It binds to the methionine sequence. So tRNA is basically transfer RNA. It transfers the amino acid to the mRNA. And I know, going back to our table, in mRNA, remember, AUG codes for methionine. So wherever that methionine is, that tRNA will bind to that place first, which is why we have a MET over here, because that's representing methionine, the amino acid methionine. So that's why there's AUG, which is the methionine sequence, and UAC, which is the corresponding RNA sequence. So it'll move from these different sites, so the EPA sites, and it will basically, in order for it to move from these sites, GTP is used, and yeah, GTP is used to power this process, you could say. And so basically, first tRNA binds to the matching codon in the A site, and tRNA binds to the matching codon in the P site, and then it moves to the A site, where then the next following tRNA will bind in the A site, in the P site, once the original one is in the A site, and then the attached amino acids will be strung together and will join, and the mRNA shifted forward, exposing a new codon, and then this tRNA is the amino acid that is attached to this tRNA will get deattached and will be attached to the one in the P site. So basically, the amino acids are being brought by the tRNA, and one tRNA will remain there to anchor all the other tRNAs, all the other amino acids to basically join together and finally form that polypeptide chain. Okay, so here's a clearer picture, I guess you could say. So the stop codon comes. So again, the termination, or the last part of translation, is when the stop codon comes. The ribosomal subunit approaches the stop codon. And so one thing to note here is that the mRNA is the one that's moving, right? The mRNA is being pulled through the ribosomal subunit. And so when UAG, UAA, or UGA, this sequence or this codon is met, then a release factor will attach or bind, and it will free the polypeptide. It will cut off any more amino acids from attaching to it. So when this this release factor binds, the polypeptide is released. The tRNA that had all the amino acids attached to it is also released, and the ribosome is broken up. Right, the large subunit and the small subunit are taken apart. The release factor is released, as well as the mRNA. Okay, so now talking about mutations. Mutations are kind of the hallmark of why we have natural selection, right? Mutations allow for change, and they allow for diversity, and they allow for evolution. So what are mutations? Mutations are basically when a mistake happens. A mistake meaning the DNA is not transcribed perfectly, or the RNA is not transcribed perfectly or translated perfectly, and there is a different product that occurs, or a mutant. And so sometimes this mutant can be effective. This mutant can be, or it can be defective. It can cause problems and serious illnesses like sickle cell disease. And sometimes it doesn't have to be that harmful, but it can have symptoms. So the results of mutations are on a large scale, and they range from really drastic to very silent. But yes, they do exist. And here I'll not really go that deeply into mutations, but I'll talk about point mutations and frame shift mutations. So point mutations are the types of mutations when there is a change in a single nucleotide. So only one nucleotide is being changed. Okay? So an example of point mutations are substitutions. And it's when instead of a thymine, maybe a guanine attaches. Or instead of an adenine, maybe a uracil attaches. Or something like that. Anything can go wrong, right? Point mutations are generally not considered to be that disastrous when you compare it with frame shift mutations. What are frame shift mutations? Frame shift mutations are serious problem makers because they change the entire sequence ahead of them. So what they do is that by messing up not only the three nucleotides they are incorrectly transcribed for, it moves the entire mRNA back or forward. And so this changes the entire set of amino acids that are yet to come or have already come to something different. So basically frameshift mutations can cause problems in like the completely other side of the strand. And it's because they shift the frame. They shift the reading frame. So for example, insertions, deletions are nonsense. So when I give you an example, it probably might make more sense. So for example, what if an extra nucleotide is added? So those are called insertions. And when insertions occur, the entire reading frame is shifted because now the mRNA is going to start being read from that insertion, from that one. So it's going to be plus one, plus one, plus one, plus one, plus one, and it's going to lead to this completely different set of amino acids, which can't possibly join together and form the same polypeptide protein they were supposed to form before the mutation. And just like insertions are a deletion, so when an amino acid is taken, when a nucleotide is taken away, and then nonsensical frameshift mutations are when are kind of when it doesn't really matter. So like there's no frameshift, but one amino acid is missing. And silent mutations or silent substitutions are when the substitution will lead to the same amino acid end product, and it's because multiple amino acid codons can code for this. Multiple nucleotide triplets can code for the same amino acid. And going back to that table, see, CCU, CCC, CCA, and CCG all code for proline. So if UCAG, if any of these third codons mess up, it's kind of okay because the end product, we're still getting proline. Right? Okay. Okay, this is just to review kind of a summary of what I talked about today. So transcription, RNA processing, and then, so transcription is with DNA, and then the mRNA is formed from the transcription, the RNA transcript. In RNA processing, the RNA transcript is modified so that it can survive the conditions outside. It is more flexible and it is more ready to be translated, which is the next step. In translation, it binds to a ribosome, and tRNA comes in and helps out by attaching the amino acids and helping them form a polypeptide sequence, and then eventually the ribosome dismantles. And again, this is all occurring in the cytoplasm, so all the amino acids join together and form a protein. A process that we didn't really discuss was amino acid activation, but it's basically when like the tRNA synthetase and the tRNA as well as the amino acid are activated. Okay, and that'll be it. Thank you so much for listening. And yeah, this is the Kahoot link. Thank you.